So there are two different aspects of molecular heterogeneity that people have looked at over the years. Initially, a number of years ago, there was a lot of investigation into the RNA-based transcriptional subtypes that seemed to be aligned with cells of origin. And the reason this was interesting is it was associated with differences in outcome. So that's something people have looked at historically. And more recently, we've looked at DNA-based genetic heterogeneity and identified discrete clusters of DLBCL that seem to be driven by combinations of genetic alterations and perturbation of pathways. This is interesting for a couple of reasons. One is that the outcome associated with these different molecular clusters is different, and it provides some additional insights into heterogeneity that is not captured just with the earlier RNA-based transcriptional signatures. And the other reason it's interesting is it provides some insights in terms of how you might think about targeted therapy. So certain genetic signatures that are associated with perturbing certain pathways really suggest targets and targeted agents that might be selectively beneficial in subtypes of the disease, but not others.